Hello everyone, Lauren Bean here. This is a lecture over chapter 11, which covers the nervous tissue. So we're going to be talking about this tissue in reference to the brain, the spinal cord, as well as the nerves which innervate our body. And collectively, the nervous tissue consists of neurons and neural glial cells, and the quantity of those is quite large, about 86 billion neurons and around 85 billion neural glial cells that support the complex neurons in our body. First, I want to provide a general idea of how the nervous system functions, and it comes down to three key things: sensory input, integration, and then motor output. So we have many different sensory receptors all throughout our body that detect changes in our environment or are activated by stimuli in our environment. And when the receptors are stimulated, it creates action potentials or nerve impulses that travel along afferent pathways to the brain and spinal cord. Within the brain and spinal cord is where integration occurs. So it brings together information, it interprets sensory information, and decides what to do with it. Should we just store the information? Should we think about it? Should we react to it? And if our brain decides to react to it, there's going to be a motor output produced. Right? Nerve impulses are going to be sent along efferent pathways to effector cells, whether it be a muscle or a gland, to cause an effect. Right? So, say we look at a glass of water, we think, oh, wow, I'd like to take a sip of that. Well, the sensory input through our visual system will occur, integration will take place, we'll decide, I do want to take a drink. Motor output, we're going to move our body to take a sip of the water. So we're constantly, using this concept at all times, making decisions in our world, right? Thinking about things, carrying out motor output. Our nervous system is divided into the central and peripheral nervous system. So the CNS includes the brain and spinal cord. This is where integration occurs, also control of motor output. Peripheral includes cranial nerves, spinal nerves, as well as ganglia, collection of neuron cell bodies located close to the spinal cord. These are little areas of processing, synapse between one neuron and another. So, chapter 12 will be all about the central nervous system, 13 covers the peripheral nervous system. We find with the peripheral nervous system, it's further divided into a sensory afferent division as well as a motor efferent division. With this motor branch here, we can divide that into somatic and autonomic. Somatic refers to voluntary control, voluntary motor fibers conducting impulses to our skeletal muscles. So conscious, voluntary motor control. Opposed to the autonomic nervous system or the ANS, that's all about involuntary motor nerve fibers, involuntary control to our smooth muscle and cardiac muscle. Falling under the ANS is our sympathetic and parasympathetic division. Sympathetic activates our fight or flight response, versus parasympathetic regulates our resting and digesting state. And we'll utilize chapter 14 to cover the entire autonomic nervous system. So the main topic of chapter 11 is the nervous tissue. So that's what we're going to start to focus on. It consists of neurons and neuroglial cells. Neurons are much larger, they're excitable cells, they're the ones transmitting electrical signals. But then there's just as many supportive cells wrapped around the neurons, wrapped around axons, etc. We find a unique set of cells for each division. So the CNS includes astrocytes, microglial cells, ependymal, and oligodendrocytes, versus the peripheral nervous system is really narrowed down to the Schwann cells and satellite cells. We'll discuss the function of each of those when we get to it. We're going to start by covering the features of a neuron first. So here's a generalized view of a neuron. We'll identify all of its parts and pieces, but first we want to think about the general characteristics of a neuron. We consider it to be the structural and functional unit of nervous tissue. They're very large, very specialized cells. They conduct or transmit or send electrical impulses or nerve impulses from one neuron to the next or one neuron to a muscle cell. Once we reach maturity and our brain tissue has all formed, right, and our spinal cord has reached its mature length, those neurons have to survive for our entire lifetime. Neurons are amitotic, they cannot replicate, so they cannot replace themselves, they can't replenish nervous tissue. What we have is what we get. So if there is injury to the neurons, that could be irreversible. Neurons are highly metabolic. They consume a ton of energy, a ton of ATP. In particular, they like to use glucose for metabolism, right? So glucose is their primary fuel source for ATP production. We'll focus on main cellular processes, which includes the dendrites as well as this long axon. We'll start with looking at the neuron cell body, or what we call the soma. This is the biosynthetic center, meaning it makes stuff for the cell, synthesizes proteins, membrane structures, neurotransmitters, right? Just making all the stuff that a cell needs to maintain life. There are some specialized organelles within nasal bodies, neurofibrils, just really specialized names referring to the organelles within a neuron that make them look different than other cells. So nasal bodies, there's dark staining aggregates of rough ER. So there's a lot of rough ER, a lot of ribosomes making a lot of neurotransmitters within the neuron. Neurofibril is just a fancy term for the type of intermediate filaments within a neuron. And then two important terms here I want you to consider is nuclei and ganglia. Nuclei refers to a collection of neuron cell bodies in the central nervous system versus ganglia as a cluster of cell bodies in the peripheral nervous system. And you'll need to consider that terminology as we move forward into chapter 12, 13, even 14 and 15. That terminology we used a lot, and I want you to automatically think, okay, collection of neuron cell bodies. Dendrites, that term, dendritic means highly branched or like a tree trunk appearance. And you can see it looks like a bunch of tree trunks with many branches. This is the input or receptive region of a neuron. It conveys incoming messages toward the cell body, which then may spread along an axon. So it's kind of this left to right flow of information through the neuron. Or if we think of this neuron as a sensory receptor, maybe a touch receptor out in the skin, well, the stimuli is first going to activate this area of the neuron. The axon is the output region of a neuron, transmitting nerve impulses toward its axon terminals. The axon is much longer than the dendrites, usually just one axon here, a little bit more specialized as well. So we can identify an axon helix, kind of this cone-shaped region where it's transitioning from the soma to the axon. As we saw here at the end, it branches and forms these bulbous swellings or axon terminals. And then along the axon are Schwann cells here in blue, providing what we call myelination along a neuron. Schwann cells in the peripheral nervous system, oligodendrocytes in the central nervous system. And there are gaps in between here, which we call the node of Ranvier. Those structures become important when we think about conduction speed of action potentials along an axon. So I like this cartoon here. So here's our dendrites, here's our cell body, here's the axon, right? So receptive region, processing, then message output along that axon. We want to consider axon transport, and this is referring to the components within the neuron and how they move and distribute throughout the cell, because these are really long cells. The products are going to be made in the cell body, but then they need to be distributed. So we can refer to anterograde or retrograde being the two types of movement. Anterograde is toward the axon terminal, retrograde is toward the cell body and away from the axon terminal. So running in opposite directions here. With this image, we're considering, say, a neurotransmitter being packed, or a collection of neurotransmitter being packaged in a synaptic vesicle. That vesicle is going to be produced in the cell body, travel anterograde to the axon terminal, hang out there, maybe be stimulated to release. That empty vesicle will then travel back retrograde to the cell body to fill up again with more neurotransmitter. Something unique here is that pathogens will exploit this retrograde transport and infect neurons within the body. So things like the tetanus toxin, herpes virus, rabies, even the polio virus, that's how they gain entry to a neuron. They'll enter at the axon terminal, just hitch a ride along these vesicles, retrograde to the cell body, and just infect the entire neuron. Viruses will even take over the cellular machinery of the neuron, start to produce more virus particles, right? So that's really how the infection occurs there. And we usually see a delay between infection point and onset of symptoms. And that's because these pathogens have to migrate up these really long neurons and then hijack the cell body. I mentioned that the Schwann cells produce myelination along the axon, specifically when we're talking about the peripheral nervous system. And this gives us a visual of how they do that. So each Schwann cell just wraps itself around the axon many, many times, just like if you took electrical tape and wrapped it around a wire. This electrically insulates the axon, increases the speed of action potential transmission, right? How quickly a message is sent. So myelinated axon fibers are much faster than non-myelinated. A similar thing occurs in the central nervous system with oligodendrocytes, but structurally it's a little bit different in how that's carried out. Other important characteristics of axons. Terminology here we want to think about is that very long axons are usually called nerve fibers. So the fiber is just implying that it's really long. Some axons are over a meter long or over 3 feet in length. So these cells are quite unique in how long they can be. So fiber is commonly used to refer to them. Two important terms here referring to bundles of axons. We use the term tract when we're talking about the CNS, so the brain and spinal cord, versus we use the term nerve when we're thinking about a bundle of axons in the peripheral nervous system. So all of our nerves, in our arms and our legs, innervating, muscles, those nerves are actually just a bundle of axons. They are not containing neuron cell bodies or dendrites. So the neurons are highly, highly organized in the body. Additional terms here are structures, axon collaterals. So when we look at our visual here, right, here's the cell body, dendrite, here's the main axon here. And what we're visualizing are these major branches coming off of it. Those major branches are called axon collaterals. That really increases connections between neurons. It produces more axon terminals, more points of contact with other cells, right? So that really increases communication points. So each one of these terminations are axon terminals. If we think of a typical neuron, it's going to have around 7,000 axon terminals. And then some real specialized cells like Purkinje cells in the cerebellum can have as many as 200,000 axon terminals or points of contact. So when you do the math, that can produce up to 100 to 1,000 trillion different synaptic connections in the nervous system. So it is such an elaborate network and circuitry of communication amongst neurons. So a synapse is just a point of contact between a neuron and another cell. So it may be a neuron contacting another neuron, maybe a neuron to a skeletal muscle cell or smooth muscle cell, or a neuron to a set of glands, right? So there's always a presynaptic neuron and some postsynaptic cell, whether it be a neuron, muscle, or gland. One specific type we talked about in chapter 9 was the neuromuscular junction, right? Motor neuron innervating a skeletal muscle cell. So synapses are going to have some similarity in the components that are there. When we think of neuron to neuron, we say there's a presynaptic neuron and a postsynaptic neuron. And next, we're going to talk about the differences between electrical and chemical synapses. Electrical synapses are less common than chemical synapses. And with an electrical synapse, there's a very close fit connection point between one neuron and the next. And there are gap junctions joining these cells together so that ions can just diffuse right between one neuron and the other. So this enables for really, really fast communication. So not very abundant, but are found in certain areas of the body. So some brain areas involved with eye movement, areas involved with the hippocampus, so learning and memory, in which we need a quick level of communication. So we find electrical synapses in those areas. Chemical synapses are kind of the standard synapse. So very common. You have a presynaptic neuron and some postsynaptic cell, and there's neurotransmitter being released. That's why they're called chemical synapses. Neurotransmitters can have either an excitatory or inhibitory effect. When we looked at the neuromuscular junction, we were just talking about excitatory effects with acetylcholine binding to the skeletal muscle cell. But it's important that you understand it can either be excitatory or inhibitory. It can give a yes or a no. When we think of the components of a chemical synapse, I hope you're already comfortable with this terminology, but the synaptic vesicles are just the sacs containing neurotransmitter. So they're stored in the vesicle and then triggered to be released when an action potential spreads down a presynaptic neuron. The cleft just refers to the space in between the pre and the post. That's where the neurotransmitter diffuses. The post will have receptors for the neurotransmitter to bind. So communication at a chemical synapse, we're going to have an action potential spread down the presynaptic neuron. That's going to open these voltage-gated calcium channels in the axon terminal. Calcium flows in. Calcium triggers exocytosis of neurotransmitters from the synaptic vesicles. Neurotransmitter diffuses into the cleft, binds to the receptor, and then causes the effect in that postsynaptic neuron, either excitatory or inhibitory. We want to consider functional types of neurons, so we can categorize them as sensory afferent neurons, transmitting that sensory information to the brain, interneurons, which shuttle information throughout the brain and spinal cord, and then motor or efferent neurons, transmitting information away from the brain and spinal cord to do work, to stimulate an effector cell. So correlating along with that are structural types of neurons: bipolar, unipolar, or multipolar. These are classified by the number of processes connected to the cell body or the soma. So bipolar, we see 1, 2 processes. These are relatively rare, but they are specific to our special senses. So vision, smell, taste, hearing. That's where we find bipolar shaped neurons. Unipolar neurons, there's only one process and then it quickly branches. These make up most of our sensory neurons and are mainly found in the peripheral nervous system. And then our multipolar neurons, kind of the standard looking neuron, generalized view of one. These are the most abundant, major type of neuron of the central nervous system and make up most of the interneurons and motor neurons. So think about these highlighted points that I have here. Now that we've identified the components of a neuron, we've talked about different structural and functional types, we need to think about neural circuitry, which is referring to the interconnected networks of neurons, which work together to process sensory input, to coordinate muscle actions, to generate reflexes, to enable complex cognition, right? How do these neurons work together to carry out all the functions that our nervous system provides? One type of neural circuit is called diverging circuits. And this is when you have one neuron providing input or sending information to many other neurons. So you see how it's kind of spreading out along this chain of neurons. It creates an amplifying effect. The signal is spreading out amongst many pathways. And diverging circuits are common when we think about motor neuron pathways and activation of large muscular groups. And it illustrates how one single motor neuron can activate many skeletal muscle fibers, right? Due to this spread of information, due to the connection of these neurons creating a diverging circuit. Versus converging circuits, we see multiple input points, multiple presynaptic neurons converging onto a single postsynaptic neuron. This allows information from different sources to come together, right, and kind of create a cohesive whole. This is really important for integrating sensory information, right? So if something is in our environment, right, someone just cooked a nice warm meal, we see the meal, we smell the meal, we may be hearing that person cook it in the kitchen, and that's a lot of different types of sensory input. So we're going to have many different neural pathways which are going to come together and converge on a common point in the brain so that we can understand and interpret the fact like, oh, okay, mom just made dinner and I'm going to go sit down and eat. Right? So it plays that really important role in sensory integration. Reverberating circuits are when you have a chain of neurons that are just sending a continuous message. So it's like a continuous feedback loop. It reverberates. This is for controlling very rhythmic patterns in the body. So breathing rate, maybe sleep and wake cycles, day and night cycles, repetitive motor behaviors. So walking, typing, not that you're walking and typing continuously, but during that period of time in which you're carrying out that motor output, you need that kind of reverberating message being sent. Then our last example here are parallel after discharge. And this is when you have one input neuron sending messages to several other neurons, which then, kind of integrate, think, connect different areas of the brain, and then it all converges back onto one output neuron. These are important for complex problem solving as well as decision making. So you think of, maybe a mathematical problem. So it starts with a question being provided, right? You have this one input, this is what we need to think about. That information kind of spreads, it makes connections with other areas in the brain, and then, we figure out that problem and we come to a conclusion. We come to a determination point, and that motor output is then determined at that point. So we can apply this concept to, like I said, a math problem, but also, decision making, thought processes that we have when we ponder on a concept, right? This is the thinking area, this is the output. Here's the decision being made. Astrocytes are the most abundant and most versatile neuroglial cell in the central nervous system. As we see from this image, here's the astrocyte with its many cellular processes. It has these flat feet that extend out, wrap around a neuron, and wrap around blood capillaries, tiny blood vessels in the brain. So it structurally contributes to form the blood-brain barrier, also provides the exchange of nutrients and waste between the neuron and the blood capillary as well. So it provides that extra level of control. There's only certain chemicals that can pass through this blood-brain barrier and affect the brain. There's many other functions that the astrocytes provide as well, regulate the chemical environment, guide young neurons during development, development of an embryo in the uterus, can also even respond to nerve impulses and neurotransmitters. So they modulate a lot of communication amongst neurons and many, many other functions. But sticks to the real key characteristics here. Most abundant neuroglial cell informs the blood-brain barrier. Microglial cells are the immune system cells of the CNS. So they function as what we call resident macrophages that can phagocytize pathogens and cell debris. That means they can eat and engulf stuff, right? They can even engulf pathogens if they get into the CNS, right? Clean up cell debris, myelination, whatever is needed. So the count of these cells is elevated during injury or infection of the brain and spinal cord. And that can be confirmed by doing a spinal tap, collecting cerebrospinal fluid, and measuring the microglial count in there. That can confirm that injury or infection has occurred in the CNS. Ependymal cells are responsible for producing cerebral spinal fluid within the brain. They're found within what we call brain ventricles. I'll talk more about that in chapter 12. But these cells are cuboidal in shape. They have cilia as well. The cilia help to sweep and move the cerebrospinal fluid throughout the nervous tissue, right? But for now, think about how these help to produce cerebrospinal fluid. Then oligodendrocytes, these are the cells in the central nervous system that produce myelination. They are highly branched. Oligo means few, dendro means branch, cyte means cell. So it's a cell with many branches, extends its cell membrane, wraps around axons, and forms the myelination in the brain and the spinal cord. In contrast, in the peripheral nervous system, it's the Schwann cells providing the myelination. Schwann cells can also help to repair damaged axons and help regeneration of an axon fiber. This is only true within the peripheral nervous system in which regeneration is possible. Regeneration cannot occur within the central nervous system. Oligodendrocytes will actually inhibit regeneration, and that is a protective barrier against tumor formation in the CNS. Then lastly, we have these satellite cells shown in purple. They surround the cell body of neurons. They help to provide structural support, some chemical support. They're really just kind of there providing a helping hand for those neurons in the peripheral nervous system. So now we're going to consider the electrophysiology of neurons, and we're going to start by thinking about the resting membrane potential. This is the same concept that we covered in chapter 9 when we thought about muscle physiology. And this just states the fact that excitable cells have the ability or the potential to do work at rest if stimulated. And the reason that they have the potential is because neurons are polarized or have a negative internal charge at rest. Right? So the inside of the cell or the cytoplasm is negatively charged, extracellular fluid is positively charged. We also find a unique distribution of ions. Sodium is the chief extracellular cation versus potassium is the chief intracellular cation. So there's this unique distribution here across a neuron as well as across a muscle cell. This polarity, this difference is what creates voltage. It's what creates the potential to do something. The sodium-potassium pump is one that's creating the unique distribution of sodium-potassium. Every time that this pump functions, 3 sodium are pushed out, 2 potassium are pulled in. So that's creating that difference in ion concentration. That creates a very steep gradient, which can be utilized if a channel opens up. That's what's responsible for causing, depolarization and repolarization. The unique thing about the sodium potassium pump is it consumes about 70% of a cell's energy at rest. That's a tremendous amount of energy being consumed just to provide the potential to be activated. Ion channels are classified as either chemical or ligand gated channels versus voltage gated channels. And these channels open up due to different types of stimuli. So, with a chemically gated ion channel, a neurotransmitter or some chemical has to bind to the channel to cause it to open up. Opening a channel is then going to allow for the diffusion of ions, flowing in its adequate direction depending upon concentration gradient. So, these are opening up in response to neurotransmitters binding. Right? We think of the neuromuscular junction. We have acetylcholine binding to the receptor, opening the sodium channel, depolarization occurs. So these chemically gated ion channels are common in the dendrites, common in the cell body, responsible for establishing what we call graded potentials. In comparison to voltage gated channels, these open in response to changes in voltage across the membrane. So when a little bit of depolarization is occurring, that's enough to open these voltage gated ion channels and encourage even more sodium to come in and fully depolarize a cell. So voltage gated ion channels are associated with full action potentials being generated within a neuron. And I'll come back to this idea when we compare and contrast graded potentials versus action potentials. Changes in membrane potential are triggered by some stimulus in a neuron's environment. And we can identify types of changes in membrane potential as either a receptor potential or a postsynaptic potential. Either way, that change in the environment, it opens ion-gated channels, allows for a flow of ions into or out of the cell, and that in turn may or may not trigger an action potential, right? But that change is used to kind of integrate and send information from one neuron to the next, or one neuron to another cell. Receptor potentials occur when a sensory receptor is excited by a stimulus. So maybe we're thinking of, our eyeball, we're thinking vision coming in as the stimulus, right? That information is being sent, but it's light coming into the eye, which is causing a receptor potential. In contrast, a postsynaptic potential is talking about the effect that occurs on a postsynaptic cell. So when you have a neuron-neuron synapse here, we have a presynaptic neuron communicating with a postsynaptic neuron. When there's an action potential generated in the pre, it will cause neurotransmitter release and activation of the postsynaptic neuron. That neurotransmitter may have an excitatory or inhibitory effect on the postsynaptic cell. So it can vary depending upon neurotransmitter type as well as receptor type. That can lead to either depolarization or hyperpolarization. So both a yes and a no message could be sent to that post. When we talk about changes in membrane potential, we classify them as depolarization, repolarization, or hyperpolarization. You need to have a good understanding of the term. You need to understand how it affects polarity, what ion causes it, right? And what it really means for the neuron. When we look at a graph of potential, on the side here we have millivolts, time on the bottom. When we look at this reading, at the bottom here, right here, it'd be rest, the resting memory potential, negative 70 millivolts. So when a neuron is not being stimulated. And then if a stimulus is applied, it causes depolarization. The depolarization is due to sodium ions flowing into the cell, making it less polarized or less negative. Action potentials peak out about positive 30 millivolts. Following that stimulus, following that excitation, repolarization will occur, bringing that cell back to rest. This is due to potassium ions flowing out of the cell. Then we have hyperpolarization. This is when the cell becomes even more polarized or even more negative due to the outward flow of potassium. This can occur immediately following an action potential, but hyperpolarization can also occur due to neurotransmitter binding and that just being the effect that it creates. Hyperpolarization takes a neuron or a cell further away from the point of generating an action potential, so it has an inhibitory effect. So we need to compare graded potentials and action potentials. And with any kind of stimulation, it starts as a graded potential. And if the stimulus is strong enough, it can lead to an action potential. Graded potentials occur in the dendrites in the soma. So when a stimulus is being applied, so say touch onto a touch sensory receptor, the changes in membrane potential first occur in the dendrites and the soma. That potential is graded, meaning it's short-lived, it's localized, it can vary significantly in its strength, it's reversible, right? So it's graded, it's just kind of isolated to this area. If the stimulus is strong enough, if you really press down on that touch receptor, the depolarization may reach this trigger zone. At that trigger zone, if depolarization occurs, we're going to find that an action potential is generated. So it's like pulling the trigger on the gun. Once you reach that point and you pull the trigger, right, the gun is going to fire. An action potential will be generated and there is no going back. So this trigger zone structurally is at the axon helix, but this trigger zone also correlates with a threshold voltage when we measure the changes in polarity. That threshold voltage is about negative 50 millivolts. So again, the stimulus has to be strong enough and reach that threshold voltage to cause the cell to fire, generate an action potential, and send a nerve impulse. Neurotransmitters initially cause graded potentials that vary in strength. And it all comes down to the amount of neurotransmitter being released, how long the neurotransmitter remains in the cleft attached to its receptors. So there's some variability there. But the more neurotransmitter released, the stronger the graded potential is. So the graded potentials occur in the postsynaptic neuron, and the effect can either be excitatory or inhibitory. So we say EPSPs and IPSPs, so excitatory postsynaptic potential. This causes depolarization in the post cell due to the influx of sodium, tells a cell to fire, produce another action potential, versus IPSBs are inhibitory, cause hyperpolarization in the postsynaptic cell, and reduce a neuron's ability to produce an action potential. Whether or not a postsynaptic neuron generates an action potential really depends on a balance of excitatory postsynaptic potentials and inhibitory postsynaptic potentials. So this area of the graph represents different types of graded potentials that are occurring on the post. The stimulus has to be strong enough, has to reach that threshold voltage to actually cause an action potential. So leading up to it can be a mixed bag of like yes and no, excitatory, inhibitory. And then again, if that stimulus is just right, just strong enough, then it will generate a nerve impulse. Summation in postsynaptic neurons occurs when you have EPSPs or IPSCs adding up in force. That adding up effect can lead to the generation of an action potential in the postsynaptic neuron. So we describe it as either temporal or spatial summation. Temporal is when there's intense stimulation by one presynaptic neuron activating a postsynaptic neuron, and that creates many action potentials. In contrast with spatial, you have stimulation by several presynaptic neurons activating a postsynaptic neuron. So with temporal, it's like one person shouting fire, 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 fire. Versus spatial, it's many different people shouting fire all at the same time. The effect is basically the same. You have that postsynaptic neuron firing, producing action potentials, sending a nerve impulse, but it's important to realize that there are different ways in which that neuron becomes activated. It's very important that you understand the difference between graded potentials and action potentials. So this table I've made here summarizes the key differences. So first with location, graded potentials occur in the dendrite and the soma. Action potentials occur in the axon. Once that trigger zone is reached at the axon helix, that's when an action potential is generated along the axon. Changes in membrane polarity, well, graded potentials, they vary. It can be depolarization or hyperpolarization, excitatory or inhibitory, all depending on the neurotransmitter or the stimulus being applied. Action potentials, this is always depolarization of the entire cell. These reach a voltage of 30 millivolts, and that is the same every single time. It reaches that voltage of 30 millivolts. For ion channels, graded potentials are due to the opening of ligand-gated channels, so neurotransmitter binding to the channel, opening it up, influx, efflux of ions. Action potentials are produced due to the opening of voltage gated channels. It's the graded potentials, the changes in membrane that open voltage gated channels at the trigger zone, at the axon, which then initiate an action potential. So it always starts with the opening of ligated channels, which will then activate the opening of voltage gated channels, encouraging even more ions to come in, encouraging even more depolarization to occur. For strength and amplitude, graded potentials, they vary, right? They are graded. Just like you can get an A or you can get an F, right? There's variability. Action potentials are the same every single time. Same strength, same amplitude, same voltage, right? They are identical in their structure. Okay? The only interpretation that occurs with the action potential is the frequency incoming to the brain. Transmission distance. Graded potentials are short distance. They are localized and they decay with distance. Action potentials are self-propagating. It's a continuous message being sent. It does not decay with time. Graded potentials are reversible. Action potentials are all or none, irreversible. Once you pull the trigger on a gun and it fires, right, there is no going back. It's the same idea with action potentials. Once depolarization reaches a trigger zone, the cell will fire. They are irreversible. Summation can occur with the graded potentials. Force can add up. The changes can add up, right? Action potentials, no summation, okay? These are the same every single time. They can't add up on each other. There's only one action potential occurring at a time. So we consider the electrical activity of an action potential. Here we're at the resting memory potential. Here's our stimulus, depolarization due to sodium, repolarization due to potassium. Here's our hyperpolarization, right? This is what an action potential looks like every single time. It maxes out at 30 millivolts. Correlating along with that are refractory periods. So these are times during or after an action potential when a neuron is less responsive or unable to produce another action potential. So during this green area here, the absolute, an action potential will absolutely not occur again. You can't have an action potential occurring on top of an action potential, right? Only one occurs at a time. This ensures that all or none event, it ensures one-way transmission, keeps it nice and consistent. In comparison to this relative refractory period, another one probably could generate at this point, but the stimulus must be really strong to stimulate another action potential because at this point it's still undergoing that repolarization and hyperpolarization zone. So only something super duper strong is going to cause that cell to fire again. Something that can vary with action potentials is the conduction speed in which they travel along an axon or along a nerve. And there's two key factors that determine how fast an action potential will spread. So one is axon diameter and two is the degree of myelination. The larger the diameter, the less resistance and the faster the impulse will be conducted, right? So the bigger the better. Degree of myelination. Myelinated fibers are much, much faster, about 30 times faster than unmyelinated fibers. So along with that structural variation, we have continuous conduction and saltatory conduction. Continuous conduction is slow in unmyelinated fibers. Saltatory conduction is fast in myelinated fibers. It's described as kind of leaping or jumping along the axon fiber, right? It's faster to hop and skip and run from one point to the other than it is to slowly walk and drag your feet. And that's really kind of the concept there with continuous versus saltatory. Myelination is good here in terms of conduction speed. So along with that, there are different types of axons, group A, group B, group C. We can identify them as either sensory or motor. So there's a lot of different subtypes there. Group A fibers, these have the largest diameter, the highest degree of myelination, so these are the fastest, right? They can conduct action potentials up to 120 meters per second. So that's really fast. This transmits sensory and motor information from our skeletal muscles, maintains proprioception, so our body position sense, allowing for very quick, precise control of our muscles. Group B kind of being the intermediate ones. So intermediate diameter, lightly myelinated, right? Conduction speeds are much less than their group A. This transmits mostly autonomic nervous system information from like our visceral organs, stomach, heart, things like that. That information needs to get where it needs to go relatively quick, but not as fast as the A. Then we have group C. These are the smallest diameter and they're the unmyelinated members, so very slow conduction speed here. Transmits information like pain, temperature, some autonomic functions. Like it needs to get to the brain, but it's not as crucial. The central nervous system, the brain in particular, differentiates between weak and strong stimuli, all based on the frequency or the number of action potentials per second, right? So it creates really like a coded message sent to the brain. And the best way to explain it is to look at an example. So, say down below here, we're looking at a touch sensory receptor and we're comparing gentle touch from more pressure or a deep type of touch being applied. Gentle pressure is going to cause, minimal changes in membrane potential, generate a lower frequency of action potentials. So it's like touch, touch, touch going to the brain. Versus if more pressure is being applied, that's going to generate more action potentials with a much higher frequency. That's like touch, 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 ouch, 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 right? Has all to do with the frequency of incoming action potentials that allow for interpretation of incoming messages. Now we're going to move on to talk about neurotransmitters, which is the last topic of this chapter. So you'll see I have a flow chart here kind of categorizing the different neurotransmitters, and we'll look at each group here and I'll break down what I really want you to focus on. Neurotransmitters are the chemical messengers of the nervous system. They can vary in their actions, they vary in the effect that they create. So they can either be excitatory or inhibitory. That has to do with the nature of the neurotransmitter itself, but also has very much to do with the receptor type that is present. Neurotransmitters have a direct or indirect effect. So, direct meaning the neurotransmitter binds and directly opens ion channels, directly causing depolarization or hyperpolarization. Indirect is when neurotransmitters act through more of a intracellular second messenger system. Usually it's a G protein pathway. Not going to dive into the details of that, but it generally creates a broader, longer lasting effect because it changes the cellular machinery inside. Then we also have neuromodulators, which are another kind of group of messengers that don't directly cause excitatory inhibitory effects, but in turn really strengthen synaptic transmission. They can influence the synthesis, release, and degradation of neurotransmitters. So playing a role in like how long that neurotransmitter is available to the postsynaptic cell. We'll start with acetylcholine, which is the first identified and best understood neurotransmitter. It's released at the neuromuscular junction and causes an excitatory effect in the skeletal muscle. Uniquely, we find that it creates an inhibitory effect in the cardiac muscle due to the fact that there's different receptors being found. And it's kind of important that there's this distinction here in separation and function between these two different muscular groups. We don't want any confusion to occur in the heart. So hopefully you have a pretty good understanding of acetylcholine at this point. You know that it opens ion channels, causes depolarization in the skeletal muscle, right? Leads to muscle contraction. Our next group are biogenic amines. So we have catecholamines and indoleamines. Catecholamines include dopamine, epinephrine, and norepinephrine. Together, epinephrine and norepinephrine are released during our fight or flight response. They're what create the adrenaline rush. They alter the body and put it into a fight or flight state. So our heart rate changes, blood pressure changes, blood goes to our brain and our muscles so we can fight someone off, right, or run for our life. Norepinephrine in particular really promotes alertness, arousal, focus, so it has more of a mental impact on that conscious state. And then with dopamine, this has to do with motor control, has to do with reward, motivation, even regulates our mood. And the reward component there is really critical for learning, pleasure, and also is responsible for causing addiction, right? So there's certain, drugs that people will take that increase dopamine levels and that creates an addictive effect over time. Then we have our indoleamines, which include serotonin and melatonin. Serotonin regulates our mood, appetite, sleep, and cognition. So, our thought processes, our problem solving ability. Imbalances are associated with depression, anxiety, and other mood disorders. So, you don't need to worry about categorizing serotonin under this indoleamine or biogenic amine category, but I do want you to think of the function of each of these neurotransmitters. And then melatonin here, produced by the pineal gland, regulates sleep or wake cycles because it induces sleep at nighttime. Depression is associated with abnormalities in norepinephrine and serotonin. Serotonin is the one we typically talk about or hear about, but norepinephrine is at play as well. And that's the one that's important for focus, alertness, arousal. Common drugs that are used for treating depression are Prozac, Zoloft, Effexor. These are selective serotonin reuptake inhibitors. So, what they do is they block reuptake of serotonin, and blocking that reuptake makes more serotonin available in the synaptic cleft. So that can allow for serotonin to continuously bind to receptors. It's more available to the receptors. So that in turn helps to increase its effect and counteract any deficiencies that may be present in the nervous tissue. Then we have glutamate, GABA, and glycine. Pay close attention here to the function. Glutamate is the major excitatory neurotransmitter in the central nervous system, so the brain and spinal cord. It really controls all processes because it creates that excitatory message. It encourages action potential formation. GABA is the major inhibitory neurotransmitter in the brain, right? So inhibitory in the brain. Inhibition is just as important as excitation, right? Always needs to be a balance of yes and no. Inhibition allows for adequate control. Also is important for relaxation, anxiety reduction, and prevention of things like seizures when there's uncontrolled excitation. And then we have glycine, which is the major inhibitory neurotransmitter, the spinal cord, brainstem, and the retina. So notice how there's kind of this division here. GABA for the brain, glycine for the spinal cord, brain, and retina. So a little bit of separation going on. Then for neuropeptides, we have substance P and endorphins. Substance P, P for pain. This neurotransmitter transmits pain signals from the peripheral nerves to the central nervous system. Antagonists of that are endorphins. These are natural opiates. They bind opioid receptors and reduce our pain perception. So there's been a lot of pharmaceuticals produced to bind to these opioid receptors and to reduce pain perception. So morphine, heroin, methadone, those are all drugs that mimic the effect of endorphins. So reduce pain, but also promote a feeling of pleasure and well-being. So highly addictive substances because they bind to those receptors. Other neuropeptides include our hypothalamic hormones, oxytocin, and antidiuretic hormone. I'm not going to focus on the function of those here in this chapter. Minimum, I just want you to understand that these hypothalamic hormones are a type of neuropeptide. These are produced in the brain. I'll spend much more time on this concept in chapter 16 of AMP2. Similarly, down here with gut-brain peptides, there's many neurotransmitters involved with the neurons in our GI tract, and many of these chemicals are playing a role either stimulating appetite or inhibiting appetite. So a couple here that I will point out, ghrelin is the major hunger hormone. So I think ghrelin to growl, making my stomach growl when I'm hungry. This really stimulates appetite. Similarly, neuropeptide Y promotes food intake. And then the opposite of that is to signal satiety or fullness. So we see like cholecystokinin, peptide YY, even glucagon-like peptide 1, GLP-1s signal satiety to control food intake. So something that's really hot on the market right now are GLP-1 medications. Ozempic, Wegovy, there's other ones out there. So these drugs mimic the effect of these GLP-1s and tell the brain that, they're full, don't need to take in any more food. So reducing appetite there helps to reduce weight and helps to counteract against diabetes and obesity. Then our last page of neurotransmitters include the purines. So we have adenosine and ATP. Adenosine works to inhibit the CNS. It promotes sleep, reduces arousal, has a very calming effect on the body. And interestingly, caffeine is what blocks adenosine receptors functioning as an inhibitor, and that's why caffeine has a stimulatory effect on the body. It prevents adenosine from binding. Last one here is ATP. So that is the molecule that provides energy for our cells, but it can also function as a chemical messenger and have an excitatory effect in certain areas. But that's not really one I want you to worry about here for the neurotransmitters. It gets a little confusing for ATP function. Okay, so that is the end of chapter 11, the nervous tissue. Next will be chapter 12, the CNS, chapter 13, PNS, and then chapter 14, the ANS, autonomic nervous system. Then we'll wrap up the semester with chapter 15, which covers our special senses.